This part of Chapter 6 will look at the general purpose of what we call the central metabolic pathways. Remember that all cells, including microbial cells, have two types of reactions in their metabolic pathways. Catabolism or catabolic reactions utilize molecules, typically carbohydrates, to break down and in the process gather energy and precursor metabolites. Anabolic reactions will use molecules from nutrients and the precursor metabolites gathered through catabolic reactions, and the energy provided by catabolic reaction to build large molecules, lipids, proteins, carbohydrates, and nucleic acids. These large molecules will be used to build cell components and to reproduce the cell. The central metabolic pathways are looked at as catabolic pathways. In these pathways, organisms will oxidize, which in biology means to rob hydrogens from carbohydrates. These catabolic reactions will provide primary energy sources for anabolic reactions. The carbohydrate that is most commonly used by these central metabolic pathways is glucose. This is a molecule of glucose. Notice that the molecule is rich in hydrogen atoms. These hydrogen atoms will be harvested to provide energy to create ATP. The key metabolic pathways that we're going to look at will be those of glycolysis and the tricarboxylic acid cycle, also known as the Krebs cycle or the citric acid cycle. A third metabolic pathway which we're not going to look at is the pentose phosphate pathway. During glycolysis and the Krebs cycle, glucose will be taken apart. The carbons will be lost as CO2, but more importantly, the hydrogens will be harvested by reduced hydrogen carriers, molecules like NADH and FADH2, which will be carry these hydrogens to the places where they could be utilized to make ATP. In the process, some water will also be created. The central metabolic pathways, glycolysis and the Krebs cycle, are catabolic pathways that will provide energy in the form of reducing power. That means the hydrogen ions, which will be carried by electron carrier molecules, mainly NADH. Oxidation reduction reactions will collect these hydrogens for respiration. Central metabolic pathways will also produce precursor metabolites in the forms of amino acids and monosaccharides. Finally, these central metabolic pathways, glycolysis and the Krebs cycle, will also provide some immediate use energy in the form of ATP. These ATP will be created during these pathways by a process called substrate level phosphorylation. Remember that in Chapter 4, we learned there were 4 basic metabolic groups: the photoautotrophs, chemoautotrophs, photoheterotrophs, and chemoheterotrophs. Among the eukaryotic organisms, including animals and plants, there are actually just 2 metabolic groups: the photoautotrophs, where we find the plants and the algae, and the chemoheterotrophs, where we find every other eukaryotic organism, including the animals. Among prokaryotes, the bacteria and the archaea, we actually find 4 metabolic groups: the photoautotrophs and the chemoheterotrophs, plus the chemoautotrophs and the photoheterotrophs. We're going to focus on that group which is medically the most important, the chemoheterotrophs. All medically significant microorganisms that cause disease, whether they'll be pathogens or opportunistic pathogens, fall under the chemoheterotroph group. Notice that in the chemoheterotroph group, we have 3 types of metabolic requirements. The organisms who go through aerobic respiration, those who go through anaerobic respiration, and the fermenters. So the central metabolic pathways are catabolic. Among the chemoheterotrophs, we have the groups that undergo aerobic respiration. These organisms will have glycolysis and the tricarboxylic acid cycle, or the Krebs cycle, as their central metabolic pathways, plus they will also have an aerobic electron transport chain. Among the groups that undergo anaerobic respiration, we will again have glycolysis and the Krebs cycle, or tricarboxylic acid cycle, and an anaerobic electron transport chain. Finally, among the fermenters, we will only find glycolysis. These organisms will use these central catabolic pathways to provide them with energy in the form of hydrogen ions that will be carried by electron carriers like NADH or FADH2. These electron carriers will take the electrons to the electron transport chain, where ATP will be produced via a process called respiration. Central metabolic pathways will also provide the precursor metabolites, amino acids, and monosaccharides via glycolysis and the TCA, or Krebs cycle. Central metabolic pathways, as we have talked about, will also provide an immediate source of energy in the form of ATP made by substrate level phosphorylation. Remember, among the chemoheterotroph, we have 4 oxygen requirement groups. Those who need oxygen and will undergo aerobic respiration will have glycolysis and the Krebs cycle and an aerobic electron transport chain to provide precursor metabolites, sources of hydrogen, and some ATP. The same will happen with those organisms who undergo anaerobic respiration via glycolysis, the Krebs cycle, or TCA, and an anaerobic electron transport chain. These organisms will utilize these pathways to procure primary metabolites, hydrogen ions, and some ATP. Those organisms that undergo fermentation, however, are different. They will utilize only glycolysis to make ATP. The Krebs cycle, or TCA, will not be utilized as a source of hydrogen ions, but rather as a source of precursor metabolites. Notice that fermenters lack an electron transport chain. We have been saying that the central metabolic pathways, glycolysis, and the TCA cycle are catabolic pathways, which is true, but not completely true. These pathways are actually amphibolic pathways. They may proceed in any direction. These central metabolic pathways can be also anabolic if the pathways are reversed. When used as anabolic pathways, these pathways, glycolysis and the tricarboxylic acid cycle or Krebs cycle, will provide large molecules like carbohydrates and triglycerides. So when used as catabolic pathways, glycolysis and the Krebs cycle will provide energy in the form of hydrogen ions, precursor metabolites, and some ATP or immediate use energy by substrate level phosphorylation. For example, glycolysis involves taking a molecule of glucose and breaking it down to 2 pyruvic acid molecules, 2 ATP molecules, and harvesting hydrogens to give to 2 NADHs or electron carriers. Thus, in the catabolic direction, glycolysis makes reducing power molecules, immediate energy molecules, and precursor metabolites. However, glycolysis can be reversed, in which case 2 pyruvic acids, ATPs, and NADHs can be used to make a glucose molecule. These glucose molecules can be put together to make larger polysaccharides like glycogen, cellulose, dextrose, or even peptidoglycan. This is the anabolic direction of glycolysis, which provides large molecules. But again, Chapter 6 is going to focus on the catabolic direction of the central pathway molecules. Especially, we're going to look at the aerobic respiration pathways, which involve glycolysis, the Krebs cycle or TCA, and finally, an aerobic electron transport chain. We will also mention the anaerobic respiration organisms, which will also utilize glycolysis and the Krebs or TCA cycle, and will have an anaerobic electron transport chain. And finally, we will mention fermentation. In this pathway, only glycolysis is used to make ATP. Fermenters do have a Krebs or TCA cycle, but it will be used to make precursor metabolites only. You should now be able to answer these questions. Name the 3 central metabolic pathways mentioned. Name the 2 most important metabolic pathways. List the 3 catabolic functions of these pathways. List the 1 anabolic function of these pathways. Which of these pathways, A through C, glycolysis, TCA or Krebs cycle, and the electron transport chain, can be found in aerobic respiration metabolic pathways, anaerobic respiration, or fermentation? Which pathway is common to all 3, glycolysis, TCA or Krebs, or ETC? Is the TCA or Krebs cycle found in fermenters? The next PowerPoints will look at these 2 central metabolic pathways, glycolysis and the TCA, in more depth.